students, in today's lecture we will continue from our previous lecture where we started talking about the importance of wastewater treatment plant for any community and for a city, and we will dive into the microbiology of wastewater treatment plant. So, when we talk about microbiology, we have three options. First, we can go ahead and we can get an overview picture of how many different kinds of microbes are present and who is eating whom, how are they interacting and what jobs they are doing. So, we might be interested in enumerating different groups of organisms, okay, and we can do it in two formats. We can divide the groups into like all bacteria as one group, all viruses as one group, all protozoa as one group, all algae and other higher orders of life as one group. So, this is one way we can do. So, we are doing it by broad phylogenetic or classification. Or taxonomy classification. The other approach that we can have when we are classifying them into groups is, instead of looking into these broad classifications, we might look at some function. For example, we might be interested in microbes that are heterotrophic, microbes that are autotrophic, or microbes that are eating other microbes, microbes that are eating BOD, COD. So, we might divide them according to the groups: microbes that are removing that are nitrifying or denitrifying, microbes that are accumulating phosphorus, and or microbes that are releasing phosphorus. So, we can classify them according to functions that we are interested in when we talk about wastewater treatment, right. The next approach that we have when we talk about microbiology of wastewater treatment plant is we can look at them in a very mathematical way and understand the kinetics of microbiology in wastewater treatment plant. So, we want to know how fast the microbes grow, how fast they eat, and how much aeration is required, how much food is required, how much biomass can be allowed, how much of the biomass can be recycled, how much should be thrown away. So, we are looking from a very practical and operational perspective. So, in this there are certain kinetics model that we have come up with: growth model, substrate utilization model, and then doing a mass balance of how much food is coming in, how much food is getting eaten up, and how much is being released. We can get equations that will help us answer these questions. The third approach that we can do when we are talking about microbiology of wastewater is we can profile the entire microbial community and learn up the names and the functions of each of the microbes. This is very tedious and it is not as informative or as targeted as the other first two approaches. The first approach gives us a very good understanding of who are the main players. And even if we do not know, for example, if I say nitrifying bacteria, I might not know all names of the nitrifying bacteria, all morphologies, all kinds of nitrifying bacteria, but I have a general idea of what they need, what environment is good for them, and what their biomass is, and what will be toxic for them, what will be not toxic for them, and I know how much time they require. So, I know the general characteristics of nitrifying bacteria. There is no need for me to know the microbes, and I know how to when I want to promote nitrification, how I can promote nitrification, and how I can control it when I want to control it. So, this is a very functional understanding of microbiology, which I think is more applied and useful, especially in context of environmental science. The second approach, which talks about the kinetics, is very important when we are actually operating a wastewater treatment plant. The third approach is just tedious, and I do not see it very relevant as for this course and for the questions that this course is trying to answer. So, in these lectures, this lecture and the lecture afterwards, I will be talking only about the broad classification of microbes in wastewater and in wastewater treatment, and then we will be talking about the kinetics: how fast microbes grow, how fast they eat, and what are the different phases of growth they undergo, what are the typical problems we come across, and how do we solve them. So, that is what we will be talking about in this lecture. So, let us get started. So, last class we talked about why do we need wastewater treatment, and these are the things that we want to get rid of in a wastewater treatment plant. Okay. So, this is a typical wastewater cycle. Here we have establishments, so we have houses and industries and or shops, commercial areas, and then each house, each commercial area is producing wastewater. At times we also collect the sewage, not only sewage but also kitchen waste, and also the storm water. So, storm water is the water that falls from precipitation, the rain, and it is a runoff, surface runoff. So, at times both of them are collected. There are street drains that collect the precipitation, there are pipes that collect waste from each of the houses, and it is all led to a combined sewer overflow treatment plant. So, here we are combining the storm water and the wastewater. Now, when this is combined severe overflow treatment plant directs it to wastewater treatment process, it treats it, and then the treated effluent is released in the river. A part of the wastewater can be reclaimed. So, reclaimed is the water has been treated to good enough standard for it to go to houses for cleaning, irrigating the grass, agriculture. So, some of the parts of the water can go back, and then the sewage produce can again be collected. Alrighty, in some places like India, we do not have direct collection of wastewater. Instead, what we have is a pre-treatment, in-house pre-treatment. So, in in-house pre-treatment, each house or each residential location would have a septic tank. The septic tank will collect the wastewater from the community or from the house, and then it undergoes anaerobic degradation. So, right here we reduce the volume of the wastewater that will go to the wastewater treatment plant, and ideally routinely it should send wastewater to the wastewater treatment plant, but this is advantageous because it reduces the burden on wastewater treatment plant. And the disadvantage in a country like India, it's a big disadvantage, is that these septic tanks require periodic cleaning, and because of the taboo in India associated with fecal matter and cleaning waste septic tanks, this is a big social problem. People do not want to clean their septic tanks, so they are making enormously big septic tanks, and then because of the unequal society that we live in, there are certain segments of our society, the underprivileged sections, that are exploited by the more powerful sections, and they are forced against their wishes to clean this manually. So, manual scavenging, it's a big issue, and one of the recommendations that I am giving in this week's homework is actually to watch a recent documentary, Kakush. It's not in English and not definitely in my native language, but thankfully it has subscript, so I encourage you to watch it and understand the problems with manual scavenging. It's not only a social problem, it's definitely an environmental problem. So, because even after we move from open defecation to toilet usage, in India we rely a lot on septic tanks. There is no one to maintain septic tanks, and the ones that are being forced to maintain septic tanks, either socially, economically, or actually literally by force, they are being exploited. So, it is a big issue, and until we solve this issue, we can't solve our wastewater treatment and our public health problem, environmental health problem also. So, this is pre-treatment for you, and there will be homework for you to watch a documentary on YouTube and answer some questions. Alrighty, ideally the septic tank here will provide its effluent to wastewater treatment plant, where it will be treated effluent will be thrown in the river, and part of it can be reclaimed. Okay. Now, in centralized wastewater treatment, there are a few very important sections. The place in the wastewater treatment where biologically waste is removed can either be an activated sludge process tank, it can be a biofilm, or we can talk about nutrient removal. These are the three places where microbial processes are used. Let us talk about activated sludge. So, if you remember in the previous class I talked about primary treatment, secondary treatment, disinfection and disposal. In secondary treatment we use aerobic oxidation by microbes to degrade waste. So, this can be of two types, as I mentioned. Either our microbes can be freely floating in the wastewater, or they can be attached to some filter or biofilm. They can make biofilm on filters. So, the attached microbes will come under this category, biofilm reactor, and the ones in which they keep moving, they will come under activated sludge. Now, here we have two options. We can have either plug flow reactor, or we can have a CSTR, sometimes referred to as SBR. And I remember in the beginning of previous class I talked about activated sludge process and SBR process. This is your activated sludge process, this is your SBR process. So, what is the difference between ASP and SBR? We will be talking about ASP very soon, but let me very briefly revisit PFR and CSTR so that you know what the difference is. I have talked about it in previous lectures, but just in case, I hope this review will be helpful for you. So, let us start with the PFR. In PFR we have a batch reactor. So, all the reactants and the products that they are forming are present together in a batch, and this batch moves from one portion to another, and as it moves from one portion to another, it undergoes certain changes. So, when it is in a settling tank, with the primary settling tank, so this is the first settling tank, whatever needs to settle will settle, whatever can settle physically in the given time will settle, and then this entire batch will move to the activated sludge process tank, ASP. Now in ASP process, this entire reactants converting into products. So reactants here is your waste, your oxygen and all other nutrients that are present. They will be eaten up by microbes and they will make products. So you have not added anything, you have not removed anything, though in ASP we aerate, so we add air because microbes will very quickly run out of oxygen. So lot of investment is made in aerating ASP, and then this batch again travels to the next step. This is secondary settling tank. Whatever can settle from here will settle, and this will be your settled, which you can refer to as product, settled solids. So notice here, this is your PFR based plug flow reactor based water treatment plant. Here the batch of your material travels from one step to another, it physically travels. So this will be another tank, this will be another tank, and the third one will be another tank, and then there will be more tanks ahead to disinfect the water, and there are tanks before it. So the key point is that this entire batch reacts. The products of this batch would be reactant of this batch. Products of this batch would be reactant of this batch, and so on and so forth. So basically it becomes reactant 1, product 1. Now this becomes your reactant, you become product, this becomes your reactant, and this becomes your product, okay, and so on. So this is a plug flow reactor, and I think talked about plug flow reactor in past and given you example of a river. So in a river or in a train, the coach does not mix with coaches before it or coaches after it. The river does not mix in the direction of the flow, but the entire cross section of river travels together. So the entire compartment or coach in the train travels together. So whatever the material is present in primary settling tank will not mix with the ASP material. ASP material will not mix with secondary settling tank material, but they will travel together. So basically it is like a river. Or like a train, and they are flowing together. So when the reactant and product reach here, they undergo primary settling. When they travel forward and they reach here, they undergo activated sludge process. When they reach here, they undergo settling tank. So by the time they are done with this train of wastewater treatment plant, you will have good quality treated effluent. So this is your PFR based system. Alrighty, now let us go ahead and look at your CSTR based system. I must say that in India initially most of our public water treatment plants were ASP based, but in past few years, in past decade, we have seen a surge of SBR or CSTR based water treatment plants for the reason that the operation and maintenance is really good and the quality of treated effluent that SBRs are producing is very nice. Also they require less space, and why they require less space, let us look into it. Okay, now let us look at what a CSTR is, what an SBR is. This is your second type of wastewater treatment plant. CSTR, the definition is continuously stirred tank reactor. So it is a tank, it is a reactor, and it is continuously stirred. So there is a stirrer that continuously stirs it. Now in CSTR, there is a continuous input of influent and there is a continuous outflow of effluent. Alright. So the influent is continuously coming in, the effluent is continuously going out, obviously because the volume is constant, the flow rate is same for both, and the reaction is happening here. So this is your typical CSTR, okay, continuously stirred tank reactor. This is different from PFR because the reaction happens in the tank. In PFR the reaction also happens here, only in the train, not in the tank. In PFR based ASP it happens obviously here, but the entire product moves from one tank to another tank, and until the reaction is not complete it won't move ahead. But in a CSTR even partially complete reactants would move. Even as the reaction is going, there is a continuous outflow of effluent and continuous inflow of influent. Now let us see what is an SBR. SBR is more like a CST, and let us see why. Okay, so your SBR or sequencing batch reactor will look like this. It is a singular tank. So unlike PFR, when you have multiple tanks for different steps and the entire batch moves from one tank to another, much like a train, the compartments of a train, in a sequencing batch reactor everything happens in a single tank. Now remember the first step of treating wastewater is screening. So the influent comes and it gets mechanically screened and then is inflowed into the tank. Once it reaches the tank, the next step is primary settlement. So this is the cycle 1. So for cycle 1 we will have primary settlement. So for certain minutes the influent will be allowed to undergo primary settlement, and then the sludge, primary sludge collected here will be removed. Okay. Now once this has been done, begins the cycle 2. Now in the same tank cycle 2 will be carried out. If you remember, the cycle 2 is activated sludge process. So the second cycle will begin after the end of cycle 1, which is primary settlement, and second cycle is activated sludge process. So here there are aerators, and the aerators will blast oxygen into air into the supernatant of primary settling tank, primary settlement process, and then the microbes will eat all the waste. BOD and COD will be consumed, how much ever microbes can eat in their cycle. So for certain amount of time the activated sludge process would be allowed. Once the time is up for cycle 2, it will come to cycle 3 or step 3. In step 3 we will have secondary settlement. So cycle 1 we did primary settlement in the same tank, and then when the times was up for primary settlement we will allow ASP for certain amount of time, and when ASP process has been done then we will allow secondary settlement, and then the biomass will settle and the secondary sludge will settle, and then this, after the settlement has happened, this sludge would be removed. Alrighty, and what is the next step after secondary settlement? Let us say it is disinfection. So the cycle 4 will be disinfection. Now disinfection can happen either in the same tank, but we do not like disinfection to happen in the same tank, because if there is residual disinfectant it will kill the microbes in the next, when this cycle is, when these cycles are repeated in the second run they will kill the microbes, so the efficiency will drop. So typically after secondary settlement the supernatant is removed and then there is a disinfection basin here. And the disinfection is done separately. So the key feature, the key difference between SBR and the PFR based ASP process is that in SBR all the processes happen in a singular tank. This is very important for a country like India where land is a precious resource, because we are running out of the land that is available for setting up expansive ASP processes, but our population continues to grow. It is very, very important for us to consider such less land occupying SBR based water treatment systems. Now the problem with SBR based water treatment system is that they require more rigorous maintenance over time. So they are more susceptible to breaking because everything is happening in the same system, and usually typically these cycles are very small. However, the advantage is that right now in India they are being operated and maintained really well, usually by under a public-private partnership, and the quality of effluent that we are getting is really good. In fact, the best in the country. Okay. Now that I hope you are clear about, and you should be clear about SBR versus plug flow based activated sludge, and then the second part is definitely settling. Either case, settling is important after degradation, aerobic oxidation by microbes. Now the next kind of wastewater treatment plants are biofilm reactors. Now in biofilm reactors, the biomass is attached to a filter or a material, and where the degradation happens, and the typical among them are trickling filters. I will show you a picture of trickling filters either in this lecture or the next. And then after this is the secondary treatment. After the secondary treatment has happened, sometimes we add a tertiary treatment to remove nutrients. When we talk about nutrients, we are talking about nitrogen and phosphorus, and we do not want them going out in the environment, and in a country like India where fertilizer use is rampant and sometimes quite excessive. It is very important to get rid of the nutrients in a wastewater treatment plant. So, after primary physical treatment, secondary biochemical treatment, we can have a tertiary treatment, and the typical way of removing nitrogen phosphorus is to alternate anaerobic and anoxic treatment. And now very important part is digestion of sludge. So, twice we have removed the sludge, one first from the primary settling tank and second from the secondary settling tank. Now, this is enormous amount of waste and enormous amount of biomass. So, it is this carbon rich, microbe rich sludge, and what do we do with this? So, it is very important for us to digest the sludge, dewater it, and then either incinerate it or use it as a fertilizer. So, this is a typical activated sludge process. This is a PFR based process, and let us quickly go through this. So, primary treatment would include receiving the raw sewage and then screening it to get rid of the big particles and particles that can destroy your wastewater treatment plant, such as aluminum foils. Then you have a comminutor which breaks them into small pieces, and then you have a grit chamber. This is where the grit is allowed to be removed. So, I did not talk about it when I said that when I talked about the basic functioning, but all wastewater treatment plants ASP based will have a grit chamber to get rid of sand and other inert particles which will reduce the efficiency of rest of the wastewater treatment plant. Then supernatant goes to the primary settlement tank of the primary clarifier. This is your raw sludge or primary sludge, and is taken away for sludge treatment. The supernatant goes then for aeration, and now in this aeration tank is where your activated sludge process happens. Look, it is connected to an air compressor. So, this will the air compressor will aerate this aeration tank. Here microbial processes happen, biomass grows very rapidly, and so this is where you have the highest microbial population. We have so many microbes and they are hungry, so they eat up all the fecal matter, they eat up all the organics here, as much they can eat, BOD and COD. And then the supernatant after clarifying, and this is, it will go to secondary clarifier, and then the biomass settles. A part of it is thrown, is rejected and taken for sludge treatment and disposal, but because this is so rich in biomass, usually the flocks and what settles in the secondary treatment plant is biomass. It is taken, some part of it is returned back to aeration tank to maintain a good amount of microbes here, initial amount of microbes. The supernatant is very clean, and it looks, ideally it is very clear, it is not turbid at all, and looks as clear as drinking water, or nearly so, and then the disinfectant is added to remove the microbes, and then this is discharged to surface water. This is your typical aeration tank, and this is your these are your secondary clarifiers. So, this is your aeration tank, circular aeration tank, this is where the aeration is happening, and then this is transported to secondary settlers. This is your trickling filter. So, in trickling filter you have these pipes, these two pipes, if you can see carefully, this bed is your filter, your trickling filter, and these pipes are throwing the supernatant from primary clarifier. So, this is your wastewater that has already undergone screening, grit removal and primary settlement, and then this is thrown on this filter, and there are my biofilms growing in this filter, and what trickles out here is your treated water. So, this then can be taken for disinfection. Trickling filter major disadvantage is the amount of the rate of treatment of water, the flow that this can accept, the loading that this can accept. The other problem is clogging. If the water is very dirty, then the filter would get clogged and the trickling will be inhibited. Now let's look at levels of treatment. Primary treatment, physical process, we remove solid. Then we can add to primary treatment, we can add secondary treatment where we are trying to remove biodegradable organics or BOD, and then we can add third degree treatment. So this is primary, this is secondary, third degree where we are trying to remove nutrients from your we are trying to remove nutrients from the wastewater. Alrighty, so in secondary treatment, so if you go back here, in secondary treatment we are trying to remove BOD beyond what can be removed in primary treatment. So in primary treatment, whatever could settle, the big particulate matter BOD has been removed, but the soluble BOD remains. So this needs to be removed, and the way we and then also removes suspended solids in the water. The basic approach in secondary treatment I have mentioned before is aerobic oxidation of biological aerobic oxidation. So we have lot of carbon, that waste material, that is organic carbon for food, it can serve as electron donor and carbon source, it is aerobically degraded, mineralized. The objective is that whatever oxygen the microbes require to degrade the waste should be made available in secondary treatment process, so microbes can consume the microbes can consume the waste as much as possible in the secondary treatment and not in the stream. So the reason why not in the stream is because if this organic carbon is thrown in the stream, microbes in the stream will try to degrade this organic carbon by using oxygen, and then they will deplete the oxygen in the stream. So the stream will become anoxic or even anaerobic, which is very bad for aquatic life, which is very bad for environmental health. So we want all the BOD to be exerted in wastewater secondary treatment. How is this accomplished? We create a very complex and rich microbial community which have amoeba, we have different kinds of microbes including bacteria, bacteriophage and diatoms and all kind of microbes, that very rich community of microbes. And this is your rich community of microbes, and this is the last slide for this lecture. And let's look here. We have dissolved organic matter, we have particulate organic matter that was not removed in primary settlement in secondary process. The dissolved organic matter will be consumed by bacteria because bacteria love to eat these small pieces of organic matter. The bacteria in turn are consumed by protozoa, which are zooflagellates or amoebas, which are again protozoa, or free swimming ciliates or rotifers or protozoa that are different kinds of protozoa. So bacteria, they don't survive very well, they are eaten by different kinds of bacteria, protozoa. Now the particulate organic matter is taken up by higher order of life, the eukaryotic protozoa, and eventually what we have is a very good protozoan strength in the wastewater. So if you look under wastewater treatment plant, you will see a very beautiful community of very diverse waste protozoa, and these are different kinds of protozoa that are present. Now if there is any disbalance in this microbial community, if the bacteria decrease or increase or decrease rather, let us say if there is bacteriophage which kill the bacteria, or there are toxins that kill the bacteria, then the protozoa would starve and the microbial community in the activated sludge process may not be able to do its job. If protozoa increase too much and bacteria are not there, then the BOD might not be dissolved, BOD might not be removed very well. So very fine balance of this is required to be maintained in a typical wastewater treatment plant. So dear students, this is all for today, where we talked about general treatment in wastewater treatment plant and the microbial community. In the next class, we will be talking about the kinetics of wastewater treatment plant, how microbes grow and how they die. Thank you very much.